So this is the phospholipid bilayer. I kind of separated this from the other ones because this is like its own stuff. But the phospholipid bilayer is the cell membrane, okay? And it's made of phospholipids. As we remember, phospholipid structure kind of looks like this, where, we have the head and then the two fatty acid chains, fatty acid tails. So this is called the phospholipid bilayer. It has 4 different names: cell membrane, plasma membrane, cell membrane, plasma membrane, fluid mosaic model, and the phospholipid bilayer. It's called the plasma membrane because all these structures, like these phospholipids, they are not stationary. They are static. They are always kind of vibrating up and down, kind of like this. So it's called plasma membrane because it moves just like plasma. And it's called the fluid mosaic model because, one, it's fluid, it moves around. And mosaic because if you search up a picture of mosaics, it has these things where it's like a bunch of geometric figures all together. It's actually really cool. I saw those in Spain. So yeah, and then it's also scientifically known as the phospholipid bilayer. So this is what the phospholipid bilayer looks like. I labeled each one, like A, B, C, and you can kind of see each one. I labeled each one, and let's go over each one. So for A, it's pertaining to this. This is called the phospholipid bilayer. And here, if you kind of zoom in, B is so these pertain to the head, and then these C are the tails. And then, okay, so B right here is the head of the phospholipid, okay? So the head right here, I drew another model. So these heads are the blue, and then the tails of the are the pink, okay? The heads are always polar, and the tails are non-polar. So here, basically, because the heads are polar and the tails are non-polar, the tails all face inward to each other because these polar heads, these blue polar heads, need to be facing the polar environment around it. The polar, for example, water. Because water is polar here, these heads have to, they're polar, so they have to face the polar environment, okay? So because these tails are non-polar, this whole area where I'm pointing to is non-polar, and we'll talk about that later, why that's important. D are these little line thingies. They are called cholesterol. Though these things may maintain the fluidity of the phospholipid bilayer, they make the membrane fluid and they aid in flexibility. They prevent the cholesterol from solidifying because they're always kind of moving. No, actually, no, sorry. They're not moving. These cholesterol, they make gaps in between the whole phospholipid bilayer, which does not allow it to freeze or solidify. Okay. E, which is the integral protein. This is this little green thingy that I'm pointing to. This is also called the transmembrane protein, and it goes completely all the way through the phospholipid bilayer. So it goes all the way through. There are 2 different types of integral proteins. One are called carrier proteins, right here, carrier proteins. Here. First, we're going to do protein channels. So the first type is protein channels, which is an example of this one. Because I told you how this in-between layer is non-polar, if we have small materials that are polar, for example, ions or water molecules, they are small and they're polar. They can't go through all this non-polar stuff. So they need to use a protein channel to give it a safe passage. Ions have individualized, specialized passages that allow them to go through the phospholipid bilayer through these like specialized protein channels. And water has specialized protein channels called aquaporin. Yeah. So, okay. And then carrier proteins, I drew a little thing about it. Carrier proteins don't look at the polarity of the material like protein channels did. It just looks at the size. So when the material is too big to go through the protein channel, and they go through carrier proteins. And this is how carrier proteins work. So this is an example of glucose. This carrier protein is built like this, like a V. The glucose goes inside. It binds right here. This is like where I highlighted in like green. This is called the active site. And it changes when it binds here at the active site. It changes the structure of the carrier protein and makes it like a inverted V so that the glucose can leave from there. And that's how carrier proteins work. So the two different types of integral proteins are carrier proteins and protein channels. Hold up. Okay. And then we have peripheral proteins, which are like these. They are also called surface proteins because they stay outside of the cell and they don't go like fully through the cell. They always like either face outwards or inwards. They face outwards or inwards. Glycoproteins are kind of like this, and they're membrane proteins that have these carbohydrate chains attached onto them. And these carbohydrate chains aid in cell-to-cell recognition and also communication. So that's the phospholipid bilayer. I would recommend getting like a blank thing and like label everything, and then also kind of like watch this video for like the functions, and then you can go back and like try to answer these questions yourself. So that's a phospholipid bilayer. I'll record other ones too.